This video will discuss some of the basics of disease modeling which are formed up in the code that is used for this lab. So the idea of disease modeling equations are basically saying, let's track how people in a population move through different categories based on how they relate to a disease. For most of these models in general, we will assume that the population is constant. People don't enter or leave the population, they just move between these groups. The simplest model for disease modeling is generally used is what's called the SIR model. And we have three different categories with respect to this disease. We have S, we have I, and we have R. We have S, which is susceptible, that is people who have not yet caught the disease, but could catch it. We have I, which could mean infected or infectious. The important thing for this setup here is infectious. They are people who have the disease and can spread it to others. And R is removed, as in they are now removed from the pool. They are no longer susceptible to disease. They cannot catch it, and they're sort of out of the picture in that sense. In the most basic model, this can either mean that they are recovered, and in some sense immune to the disease because they've recovered for it, or they are deceased from the disease. Those are both sort of lumped into the removed category. Throughout this lab, you'll see a separate model where you add a separate category of people who have recovered, people who passed away from the disease, as in two separate groups. You can see how those two different populations relate to each other. How can people move through these categories? So the main idea is that it is possible for a person who is susceptible to disease to become infectious if they catch the disease from an infectious person. So people can move from here into here, and that happens when a susceptible person comes into contact with an infectious person, and there's a certain rate at which the disease gets transferred. So this is some constant times S times I is the rate at which someone moves from S into I. Then people can move from I into R, either recovering or passing away from the disease, at some rate beta times I, some sort of decay rate on population I becoming recovered or removed. We assume in this case there's no way to go from S directly to R. You'll see a case in the lab that actually adds in that transition there. So what does this mean? Well, this means I can write a differential equation system that models this. Right, this tells me that, for instance, ds dt is negative alpha si. Scroll back up. The S box has one arrow leaving it with rate alpha si, so that comes in as a negative sign in my equation here. I could similarly write the other two. And this system here is the basic SIR model for disease spread. Now, for initial conditions, these equations here work and are sort of most accurate when all these variables represent population fractions. That is, S is the proportion of the entire population that is susceptible, I is the proportion that is infectious, and so on. With that, these equations work and can model them. What that means is then the initial conditions must be all positive, or at least all non-negative, and all add to 1. In general, it also makes sense for the initial value of R to be 0 because at the start no one's recovered yet, and the initial value of I to be relatively small compared to S because only a small percent of the population has the disease that they are spreading at the start of an epidemic or a disease outbreak. An important thing to keep in mind with these models is they all come with certain constraints on what the system is and how it behaves that may or may not be true in a given case, and it's important to be able to understand what those simplifications are when trying to see if a model is going to work for a given physical problem. For instance, if we go back to the picture, this is assuming there's no way to get from S right to R. As in, there's no vaccine for this. There's no way for someone to go right from being susceptible to being removed and not catch the disease in the process. It also assumes that once someone gets the disease once, they can never get it again. Right? Because once they land into R, they're no longer susceptible. For some diseases, it's appropriate to have an arrow that goes back this way because people after a while are no longer immune. And so there's another rate of change that way that comes into effect. You can also have more complicated diseases, things that maybe there's a portion of the infectious period that happens before symptoms happen, or maybe there's a part where you're infectious and then a part where you aren't infectious. And so you can sort of balance those two and have more stages in this process. but all of that adds more complication to the model. The simplest model is the one we started with here, with just 3 groups, and you only go in one direction between them. But normal disease and actual diseases in the real world are much more complicated than this, but you can still get some reasonable predictions and ideas out of this simple model here as well. So the code you will see for this lab is going to run this system for you. And so I'll be able to see the results, analyze them, and then interpret what the graphs are telling you based on the outputs of these models for given input situations. And the next video will go more into detail about that code, how it works, and how to adjust things to get the results you want out of this lab.